This is part one of chapter 15 online lecture video. This video is going to cover the innate immune response. The innate immune response is a non-specific response to foreign antigens. Antigens are anything that can invoke an immune response. So bacteria, viruses, fungi, food, dust, pollen can all be considered antigens. Um, the innate immune response is fast and always protecting. We're said to be born with these systems intact and ready. They include the first line defenses, which are the external physical barriers, and then second line defenses. Once these first lines are breached, the cells and different blood chemicals are there to act non-specifically to try to get rid of the antigen. Part two of the lecture video for chapter 15 will be the adaptive immune response, which is a specific response to invasion. That's considered the third line of defense, and that responds uniquely to different species of different pathogens. So here's a little overview of the immune system, the innate immune system versus the adaptive. So the innate, again, include the first and second line defenses. The first line defenses, we'll talk about the skin, mucous membranes, normal microbiota. Second line defenses, different blood cells, processes like inflammation, fever, and other antimicrobial proteins that are in the blood. The third line defense, we'll talk about the specific lymphocytes, the T cells and B cells, and antibody production with the adaptive immune system in part two video. So the first line defense skin consists of the epidermis, which is tightly packed with cells, and there's also keratin, which is a protective protein. The outer layer of the skin cells are dead, which slough off constantly, and they remove bacteria along with those dead cells. The dermis, the dermal layer, is difficult to penetrate, so microorganisms rarely penetrate skin on their own. Very few can actually burrow into the skin. So if our first line skin is going to be breached, it's going to usually be breached through a cut or abrasion. Some antimicrobial characteristics of the skin is it tends to be dry, which inhibits microbial growth. So there's a more diverse array of microorganisms that live in the warmer, moister areas of the body. The skin also contains fungistatic and bacteriostatic fatty acids that inhibit microbial growth. And it's also slightly acidic, again, inhibiting microbial growth. As we know that we have normal microbiota, which provides competitive exclusion from pathogens from actually being able to attach. The mucous membranes, these line the gastrointestinal, respiratory, and urinary tracts, and contain mucus that traps microbes and prevents the tracts from drying out. With the respiratory tract, there's the ciliary escalator that transports trapped microbes in the mucus out of the lungs. And again, the mucous membranes would also have normal microbiota that again provide competitive exclusion. So the ciliary escalator will grab dust, microbes, and other things that enter into that tract and move the trapped particles at a rate of about 1 to 3 centimeters per hour. So certain chemicals, including cigarette smoke, can damage those ciliated cells, which is where you would develop what's called the smoker's cough. Some physical defense factors of the mucous membranes would be saliva that dilute and wash microbes off the mouth surfaces, urine that flows and flushes microbes from the urethra. Some chemical defense factors of the mucous membranes include lysozyme and perspiration, tears, saliva, and urine. Lysozyme is an enzyme that will break down peptidoglycan, so gram-positive microorganisms are more susceptible to the lysozyme. We also have a low pH of the stomach acid, which very few microbes can actually survive that low pH. There's also a low pH of vaginal secretions, relatively low pH of urine, although the urine pH can vary, going from acidic to sometimes basic. There's also lactoferrin, which is iron-binding compounds that bind iron and keeps iron away from unavailable basically to microbes. So that iron is an important element for certain enzymes in their active sites. So milk, saliva, tears can contain lactoferrin. Like I mentioned, normal microbiota is very important in our first-line defenses, providing competitive exclusion. So the normal microbiota, they compete with pathogens, or they can alter the environment that prevent pathogen growth. So they take up space that prevents adherence. They can utilize available nutrients, preventing the growth of pathogens, and they can secrete toxic chemicals that can potentially inhibit or kill pathogens. The second line defense operate when the pathogen penetrates the skin or gets past the mucous membranes, and that includes different cells and antimicrobial compounds in the blood. So looking at some cells, they're non-specific. These are non-specific cells. There's granulocytes and then agranulocytes. Granulocytes contain large secretory granules, usually have chemicals in there that are involved in inflammation or phagocytosis. So there's three types. There's neutrophils and eosinophils. These are phagocytic cells, and they are capable of migrating out of the blood vessels into surrounding tissue that may be infected. And then basophils, these can leave the blood and release inflammatory chemicals. Basophils are non-phagocytic cells. High levels of these three types of cells can be a sign of infection. Agranulocytes, they lack the secretory granules. There's two types of lymphocytes. These are involved in the adaptive immune system, including B and T cells. Monocytes, which leave the blood and mature into macrophages. And macrophages are phagocytic machines. They'll take care of a lot of the foreign antigens. And then lastly, dendritic cells, which are very important in the immune system. They're found beneath the skin and in the mucus, associated with the mucous membranes, and they sample antigen, and they'll take it to local lymph nodes to present the antigen to adaptive immune cells. So these are what I call scout cells. They're constantly sampling antigen. This schematic shows how these cells arise in the bone marrow, and they differentiate into different types of cells depending on where they mature, where they're found after that. So we have the myeloid stem cells that can differentiate into plasmids or platelets, basophils, neutrophils, eosinophils, and monocytes. And as you can see here, these three are involved in phagocytosis. The lymphoid stem cells, they mature into the lymphocytes. B cells mature in the bone marrow, T cells in the thymus. But again, those lymphocytes, those are involved in the adaptive immune system. Looking at some antimicrobial compounds of the blood, some proteins that are important, there will be complement proteins, interferons, and then antibodies, which we'll talk about in the second video. So complement proteins are a series of proteins, numerous proteins that circulate the blood in an inactive form, and they can be activated by these proteins binding to microbial cell surfaces. They can also be activated by antibody-antigen complexes. So when an antibody attaches to its complementary antigen, it can stimulate the activation of complement. The effects of complement activation include opsonization, which is enhanced phagocytosis by attracting phagocytes. So very much like an antibody attaching to an antigen causing enhanced phagocytosis or flagging, that term is referred to as opsonization. Complement can also trigger inflammation, and a full activation results in what are called membrane attack complexes, or MACs, which would result in the lysis of the foreign cell. So here you see all these little pock marks on the bacteria here. Those are all membrane attack complexes, so the cell membrane would be compromised and the cell would be lysed open eventually. Interferon, this is released by host cells that are infected by viruses, and the interferon may actually cause many of the symptoms associated with viral infections. And what interferon does, so if you're an infected cell, a gene is activated to produce interferon, and that interferon is released into the surrounding environment. and that causes the neighboring cells to produce what are called antiviral proteins that inhibit viral replication. So they can degrade messenger RNA and bind to ribosomes and prevent viral replication. So here, if we have a virus infect this cell here, again, in the nucleus, we would have a gene activated to produce interferon in orange here. This interferon is released into the extracellular matrix. So as time passes, here, this is the infected cell to the right, this virus is going to replicate and then be released. Okay, this cell here that receives the interferon signal then produces antiviral proteins, AVPs here, so it's ready for when the virus actually does invade. So now as time passes, this cell here that has the antiviral proteins, as this virus infects this cell, these antiviral proteins are able to degrade messenger RNA and bind to ribosomes to prevent that viral infection from persisting. So I like to consider interferon as basically a warning signal to the neighboring cells. Inflammation is another innate immune response. This is a nonspecific response to tissue damage. So sunburn, cuts, chemicals, abrasions, any injury can cause inflammation. There's two different types. There's acute inflammation, which develops quickly and is typically beneficial, eliminating whatever condition that caused it. And then chronic inflammation, which is long-lasting and causes actual tissue damage, resulting in disease. The four classical signs of inflammation are redness, swelling, pain, and heat. So every one of us have experienced inflammation at one point or another. From getting hit in the head with a hard object, you see the signs of inflammation right away. So inflammation results in a dilation and increased permeability of blood vessels. So the innate immune system can leak out of the blood vessels and into the damaged tissue area. We also get chemotaxis, which is a migration of phagocytes. So chemotaxis, where we have the tissue damage, those damaged cells release cytokine chemicals, and the phagocytic cells go from a low concentration to a high concentration towards that site of injury. And ultimately, inflammation should result in tissue repair. Fever is the last part of the innate immune system. Chemicals called pyrogens can trigger the hypothalamus to increase the body's core temperature. So our normal core temperature is set at 37 degrees Celsius. Fever would be anything higher than 37 degrees. There's different types of pyrogens. They can be bacterial toxins such as lipopolysaccharide or other toxins. Be cytoplasmic contents of bacteria released when the cell is lyse, or antibody-antigen complexes. The advantages of fever would be triggering the production of interferon. So again, that warning signal. So a viral infection, a viral infected cell can release that. Fever is also thought to enhance the performance of phagocytes and increases the temperature above optimal for the microorganism. So if you remember from some of our lab activities in Chapter 5, bacteria have optimal growth ranges. Once you go above that optimal range, the activity of that bacteria quickly declines. The disadvantage of fever would be dehydration, body aches, fatigue, and then if your body temperature reaches 44 to 46 degrees, that can be fatal. All right, this will conclude the Chapter 15, Part 1 Innate Immune System video.